think I'll need anything here, but all right. So I think you guys know I'm a pharmacist by training. So I'm a clinical pharmacist by training. And so drugs are my jam. That's what I tell people. And I practiced for 15 years before Rory was born. So I'm very, I guess, into drugs in a completely legalized way in the state of Colorado. Which, if you don't know anything about drug laws in Colorado, they're almost as loose as the drug laws in California, but not quite. I'm going to talk a little bit about not the ones that Ilka has mentioned, but some of the other ones that have come up along the way. So, and just give you a quick introduction to just drug repurposing and what that is. And why that's such a big trend right now in rare disease is this is the path to novel or new drug development. So when you look on this column here, you can see not only how long it takes, but also how expensive. Actually, the expensive part is cut off. It's billions of dollars to develop a new drug. And if you do drug repurposing, which is essentially taking a therapeutic that already exists, that already has safety data, some efficacy data, and just trying to find an alternative use for it, including supplements, which luckily, NARS-1, it lends itself to a lot of supplements just by the nature of it being, an amino acid. And also, unluckily, because supplements can also be awful. But we'll talk about that too. The time, much shorter, right? It's about half the time. And the cost is a fraction. And that actually for us, it's beneficial because we are small. So it also means that if we can get at least even observational data or small studies, it can really help us better understand the therapeutics in a much cheaper and more efficient way. Let's see what else I got. Oh yeah, this just is like the trends. It is changing and it's happening very, very quickly. So I work in this space full time now, and so these are the things that I hear about. And AI is also like drastically changing how quickly not just drug repurposing happens, but just novel and new drug therapy. It's cutting that time down to months instead of years, which the scientists probably know that too. It's probably helping them. So just a quick recap. We did a project in 2024 with Perella, which is, you've already heard Ilka talk about yeast models a little bit. Not really, just like she just mentioned it. So we did this with them because they do a yeast model. It's very fast, it's very efficient. And they put 8,000 different compounds on their yeast models, and they really measure for rescue. So they try to see if the growth improves in those yeast models once they remove the NARS-1 equivalent. So the next steps was I had a student who just finished evaluate all of my supplements that came up on this list. And then I think our thought is, do we implement a few? Do we do additional testing? And then how do we look at measurement of outcomes? So today I'm actually just going to talk about the supplement data. There's a couple of drugs that are FDA approved that I'm also interested in that also came up on our hit list. I think I have a little bit. So here's my sciencey table. But basically, I'm going to talk about all these things in green up here today. These are all different supplements or nutraceuticals. And so I'm going to talk about the data around them. So the thing when you look at supplements and nutraceuticals, there's almost zero data that exists because they're not required to show that. It's a little bit stricter in EMA. Ingrid's like, I don't know. In compared to the US, yes. So there's a little bit more information, but this is what I had my pharmacy student do on rotation with me, is really look at the data and see what's out there. And that can kind of help us determine how to move forward. Okay, so the first one is L-histidine, actually. And Ilka mentioned HARS in her talk. And so again, and actually Ingrid and I talked about that in the summary of Stephanie's work, is that there might be some compensatory mechanisms that are happening between our clusters of genes. And so this is histidine came up on our hit list as rescuing NARS1. So histidine is another amino acid, and it has worked well in where there are variants in HARS genes to supplement. And so, oh yeah, I already did the first point. Look at me. So there's not a ton of data out there in terms of supplementation, other than in HARS-related disorders, which is great. And we have some dosing information, again, from these HARS-1 disorders. But that's the question, is can you use that dose? Can you use a different dose? What's the safety around that in variants with NARS1? Because we do know in HARS patients that it doesn't work in all the patients. So again, those are the questions that have come up as we looked at this data. But histidine is readily available as a supplement, which is a positive, because asparagine is not readily available as a supplement. It's a lot harder to find. But again, we have an element and we have an idea of like what to measure, and we have scientists who actually know about histidine, an amino acid, and as an amino acid as a function.
Histidine should be safe. There's not
Yes.
studies on the safety of histidine, so there are no safety concerns with histidine, or they will work or not.
Correct. Yeah. Lots of safety data. Yeah. Yes. Which is why it's been so successful in HARS patients too. Yeah. And then the other one I wanted to, now that I'm going to kind of go into these other supplements. And so this, they actually compared, Perella looks at kind of their entire library that they've done their experiments on. And so this is compared to FARS2, which is phenylalanine, and 2, meaning it's in the mitochondria. So it actually looks like we behave in opposite ways in a lot of cases. Which makes some sense because they're in a different part of the cell than we are. But there's a couple of things that have showed up that are similar, and I'll kind of talk a little bit about what those are and why they might be interesting to look at. I think the first one, yep. Wait, is that my first one? Yep. So FAD, flavin adenine dinucleotide for short. So it is necessary. It's necessary to produce energy. So we've talked a lot about having to have charges and make sure that we have the right environment to be able to add asparagine to the end of that protein to make that protein functional and continue to do its job. And so one of the things that in Ingrid's work we know is that charge, that amount of energy there might be reduced. So this FAD, what it does is it helps to create some of that natural energy. The downside of it is that it's not bioavailable, meaning if you were to, because you can buy this, if you were to take it, it does nothing. It just breaks down and you pee it out. So you've wasted a lot of money. Or very little of it is going to get back into your cells. So, but riboflavin or B2 is absorbable, so that's a precursor. And so if you can absorb it and get it back into the cells, it gets converted back into FAD. And so this has actually been studied in different forms of ataxia and neuropathies, and including LARS2. There's been a really small case report of mitochondrial leucine tRNA synthetase, and B2 is often a part of that cocktail in the mitochondrial disease space. So again, it's something that's very safe. It's very hard to overdose on, and it's generating a lot of that energy. So it could be something that we think think about in combination with some of these other amino acids. So again, I don't think that any of these that I talk about today will have a massive impact on their own. But if we start thinking about how they're combined, there might be some combination that might provide some, some element of relief. So the next, this actually isn't a supplement. Alpha-alanine was originally developed as a drug through depression, through different dopamine neurotransmitter modulation back in like the 70s, and never came to fruition. But it really is the active form of phenylalanine, which phenylalanine is FARS, which is another amino acid that's part of our sort of cluster of genes. So they don't really talk about why it was abandoned. I don't know if there was some toxicity there. It's actually very hard to pull these papers that have been scanned in from 50 years ago. But I think could be possibly that phenylalanine, when accumulated, creates toxicity. And so we have another disease state called phenylketonuria that people live with every day, which is a lack of the enzyme that breaks this down, that allows phenylalanine to be excreted safely from the body or allows it to break down into other amino acids. So we know what happens when you accumulate too much phenylalanine. It's very bad, and you can have neurotoxicity symptoms. But interestingly, what we do know is that the D form actually can prevent that accumulation by binding up the L. And so could be, again, this is a long ways away, but that dosing, we wouldn't know. You can get DL, which is I don't know how to explain racemic mixtures. A left and a right. Yeah. There's a left and a right. And so you have a right, the right side, your D side, which is the active form, which is the non-toxic form, and the L side, which is the left side, and it's the toxic form. So if you can isolate one without allowing it to build up, there's some potential there, a possibility there that it could provide some rescue without the toxicity. Also knowing that kids with NARS1 don't lack the enzyme that will break it down. All right, coenzyme A, there's actually not much here. So my student looked up quite a bit of this, and I think again, this is one of those things that you can purchase coenzyme A, believe it or not. All this stuff that that people waste their money on. That you can purchase coenzyme A as a nutraceutical. Again, it's immediately broken down into by B5 and excreted. So you can't, nothing. You're just consuming it and peeing it out. B5, even when they've tried to supplement it and get enough B5 in the system to convert into CoA, you can't raise the levels, it just doesn't go anywhere. So you would have to take massive amounts to actually create that amount of energy to do something with it. So B5 as a supplement, fine, it's safe, but it's probably not doing a whole lot when it comes to this piece of things. So I'm moving it off the table. I'm just telling you all the data that we did look at to get to what we have. And then the last couple ones that I'm going to talk about actually are also energy producers. And we just looked at them as a group, basically. So ATP, GTP, ITP, and CTP, which are all different amino acids that produce energy, believe it or not. So they're very rarely studied. The only thing that has been available for use that you can purchase is GTP and ATP, but there's almost no data. There is no data on GTP in the literature at all. ATP has 2 tiny little studies That's muscle mass improvement that they measured, and actually the other one is kinetic data. So it's actually looking at how the supplement, the nutraceutical, is metabolized and broken down in the body, and the effect that dosing had on it. So I'm not ruling this out, but this would be a big upside hill. Yes, Ilka.
So if I just add something.
Yeah.
We tried adding extra glucose to the cells, which gets converted.
Yep.
Didn't do anything.
Didn't do anything. There we go.
Because I think the cell just does what it does. It
Yep.
doesn't, it's not like, they don't need extra energy, the cells, so they don't convert it.
They don't use it. Yeah, they just get rid of it.
All done.
All done. Yeah. I think that makes a ton of sense to me.
We had the same thought, right?
Yep.
Was it ATP? Wasn't it?
No. Yeah.
Okay, for the recording, we tried adding extra glucose so that it would make more ATP. Didn't do anything to the cells. We tried, which means we're only trying it in cells. We're not trying it on
Right.
humans, right? It's always the difference, but.
Yeah, but again, if that's in the cell, so they're putting that sugar directly on the cell, and nothing happened. So once you introduce all of the complexities of a human, it would take a massive amount of sugar, right? And you're not going to see anything except for spikes in your blood glucose over time.
Yeah, then we get diabetes to the
That's
problems.
right. We try not to do that. No. And actually and then the last class we looked at actually was these flavonoids, or this cluster of flavonoids. And what Perella did was actually compared NARS1. They had algae 11, which is a glycosylation disorder. And this is kind of where our idea from glycosylation came from as a shared biomarker. Because when we looked at our hit list together, the rescue of the drugs was actually pretty similar, right? So the same nutraceuticals, the same supplements for NARS1 that were showing some benefit are also showing benefit in this particular disease as well. So we've talked to this group. Ingrid and I have talked to this group because they're in our grant cycle. So we're trying to track down the families, because I actually have heard that they've tried one of these supplements in their kids. So I'm just curious to know how it went and what they're doing. So my student looked at each of these flavonoids, the apigenin-based ones, and then the luteolin-based ones. And kind of this is what he found. So flavonoids are like diffusely available in our environment, in plants and what we eat and consume, and really are there largely to support anti-inflammatory effects. And so we've talked a lot about inflammation today. So again, it's trying to temper that stress response and all the other things that kind of happen in our cells. So when we looked at this, really all of the products that were tried in the NARS1 cells are only available as lab grade, meaning they're not available for human consumption or use. They're only available to test on cells in a lab and look at outcomes. And so I had my student then look at luteolin by itself and then apigenin by itself. You can get luteolin, you cannot get apigenin, even though apigenin actually is better from a consumable standpoint. It's more bioavailable, meaning more of it's getting absorbed. So we did a little bit of work on this. We don't have a lot of human-based data. We have a lot of cell-based data, and mice data. So when we've looked at the data in other neurogenic disorders, that are degenerative, so Parkinson's, multiple sclerosis, Alzheimer's, which is actually where you find a lot of data when it's related to brain stuff. They actually had some decent clinical outcomes there in terms of behavior and cognition and memory, and then some good inflammatory biomarkers. So I think, again, I would not rule this out. It's just going to be, how do you look at it? How do you test it? How do you get it a dose? And that's where the uphill battle is. And then I had my student do a feasibility score based on, we just totally made this up, but it made sense to me. But we had them kind of put together all these different supplements and then kind of talk about, all right, do we have good data, primary literature data, in cells or in mice? Do we have good data in humans, of which there's very little? And then tertiary data, meaning just like conglomerations of data, and then dosing information. Because when I think about sort of the endpoint and where we want to go, it is ultimately into individuals affected by NARS1, right? And so if you don't have good dosing data, then that's something you have to go back and you have to think about how do you get there. And then the same thing about availability or accessibility. If you can't find it, then it doesn't matter. Then you have to create it, which then gets you in the pipeline of development and cost and everything else. And then looking at safety data. So that's how we came up with our scores. Again, totally made up. And so the higher the score, right, the more sort of, the more I am more invested in thinking about how do you pursue those things. And I think that was the last thing. Oh, this is just my FDA-approved drug list that I'm very interested in that was left, which is, let me see if I can get up here. Yeah. So sapropterin is actually used in PKU. So it's a synthetic enzyme that breaks phenylalanine down naturally. And so I'm wondering if it's not the phenylalanine that does the rescue, but the tyrosine, which is the breakdown product of phenylalanine. I don't know. I don't know the answer to that, Ilka. But the fact that phenylalanine comes up and then it's essentially converse, right, on this list makes you kind of wonder, like, what's going on at a cellular level that might benefit it. Oh, I forgot. Oh, I'm missing one. Adamantine, which is actually amantadine, which has been on the market for, I don't know, 50 years. It was developed as an antibiotic, an antiviral originally, and it sucks as an antiviral. But it is used in Parkinson's to help with tremors. It has been studied in autism to help with behaviors and irritability and aggression. It is super benign. So I'm very interested in that. Nitagnolide, which is actually a diabetes drug, speaking of sugar, but really does a poor job of regulating blood sugars. And then the other one is scopolamine. So if any of you have seasickness issues or motion sickness issues, this is that little patch that goes up back here. So this is where acetylcholine, Stephanie talked about acetylcholine in her study. This is something that helps to regulate acetylcholine. So these are all sort of FDA-approved drugs that I think about, like, how do we look at those in cells? How do we look at that in terms of how they might, serve as a rescue for kids with NARS1? I think that might be it. That's it. That's all I have. Any questions? Oh, it's frozen. Knockout.
But does the knockout complement it with nothing?
Yeah, so they, say that again?
So, but the knockout,
They knocked out the D81 in one.
So it's a knockout complemented with what?
I don't think I understand.
Oh, I'll talk to you
Okay,
later.
yeah.
So they didn't look at one specific
No.
NARS mutation?
No, they did not.
Okay.
Just the full removal. Yeah, yeah.
I was just going to add, you all are asking questions to each other and laughing about, like, why does that happen? And I said, this side of the room is over here laughing like, we don't even know what you guys are talking about. So it's super complicated. I think you did a great job setting it up at the beginning of, like, basically there are drugs out on the market that we know could impact the expression of what comes out of the NARS1 gene. We know there's impacts of like the downstream things, et cetera. So trying to find things out there that already exist, don't cost money, they're already made, we don't need to spend money on that, but we just need to know whether they could impact our gene in a positive way. Is how I would summarize for this half of the room. Okay. Okay, I think now we're moving on. It's in the PowerPoint